Okie dokie. Okay. All right. So make sure that you're doing the seating chart for both the exam yesterday and for today. So make sure you do both. Look at the bottom. There's a little tab. So make sure you go in and do your both days. Okay. Minimize that. View full screen. Okay. All right. So, why did the student eat his homework? Because it was a piece of cake. No, no. I tried.
I thought it was fun.
I thought it was good. Well, y'all have been watching the news, right? So they discovered a genetic link to diarrhea. It runs in your genes. Okay. You like that one? All right, so grades will be posted later today. I'm still getting a few in, so they'll be up later today. So you can check by the end of the day, they should all be up. All right, so we're gonna start on glycolysis. So for the rest of the semester, what we're actually gonna work on is metabolism. So now that you understand all the background behind how enzymes work and all the nitty-gritty details, now we can take some individual pathways and see how they actually function within the cell, which is really awesome and exciting. So we'll talk kind of in general about metabolism first. So we'll talk about kind of everything that accounts for all of the different energy conversion pathways, right? Because when we talk about metabolism, we're not just saying we're going to break down sugars, right? That's not the only thing that happens. Metabolism is really an integration of lots of anabolic and catabolic pathways that give you your overall metabolism. So it's very complex. Then we'll talk the rest of the chapter really about glycolysis and everything you need to know for that. So when we talk about metabolism, it is all of the biochemical reactions, all of them that occur in one organism, right, that converts chemical energy to work. That's a lot of different things. Like we make that statement and it sounds simple, but it's not. It's tons of different catabolic reactions. It's tons of different anabolic reactions that have to occur in order for you to function on a daily basis. So when we talk about catabolic, what is catabolic? We're breaking things down. The reverse is anabolic. We're going to make things. So which one is going to require energy? Anabolic. Which one is going to produce energy? Catabolic, right? And so generally, what form do we store that captured energy in for anabolic pathways? We capture it and we turn it into the energy currency of the cell, ATP. We turn it into ATP. Okay, so let's take an example, right? So here's an example of metabolic flux. If you want to understand how your body regulates things, you have to understand metabolic flux. So it's how we interconvert things. So we're going to take two situations, right? We're going to say, we're going to look at our liver. It's going to be in our liver every day, y'all, every day. Maybe. Okay. The liver, right? Okay. So the liver is one of two places where you store glycogen. What is glycogen? It's sugar, it's chains of sugar, and there are long chains and there are branched chains, right? So what happens when you've been sleeping? This is before breakfast. You haven't had anything to eat since last night. Tell me about your blood sugar. It's low. So A is going to be our situation. This is before breakfast. So basically, we're going to call this a fasting state. And so what did you tell me about your blood sugar? Our blood sugar is low. We have low blood sugar. So if we have low blood sugar, are we going to be making glycogen? No, but do the enzymes that facilitate production of glycogen, do they exist in the liver at that point? Yeah, they do. They do. Everything's basically there. You're just changing which reactions we're doing based on which substrates or metabolites we actually have. So if our blood glucose is low, do we have a lot of glucose hanging out? No, we don't, but our brain needs it. So what are we going to do? We're going to take that glycogen, and what are we going to do with it? We are going to break it down. We're going to go through degradation pathways, and we are going to make glucose and ship it out to the brain. And the brain's going to say, thank you so much, right? So important. The other thing that is going to happen is at the same time, we're going to take pyruvate. And from pyruvate, we are going to create glucose via gluconeogenesis. So all these pathways are here, but we're really only going to use some of them within the cell. So if we degrade glycogen to glucose, is that an anabolic or catabolic? If we degrade glycogen, we are breaking something down, right? Now, what about the production of glucose via gluconeogenesis? This one is anabolic. Okay, so let's talk about a second situation, right? Where we've just had lunch, or we just had breakfast. I guess let's say that. We just ate breakfast. We ate breakfast. So what happened to our blood sugar? It went up. So now this is our fed state. We had a fasting state, we have a fed state. In our fed state, tell me about the amount of glucose in your bloodstream. It's high, right? Do we need to put any more glucose into the bloodstream? No, so what are we going to do? We need to take it out of the bloodstream. So there are a couple of things that we can do. We can use glucose in glycolysis, right? And so when we use it, what is that fuel for? Normal cellular functions, right? We're going to produce ATP. When we go this way, we're going to make ATP and we can run the cell. Yay, right? But at the same time, you probably still have some glucose left in your bloodstream. You don't need it all. So now what are you going to do with the remainder of the glucose? You are going to store it by creating glycogen. So all the excess glucose goes to glycogen. And there are some other pathways, but this is really simplistic. So all of these pathways are available, but we don't use them all at the same time, right? So how do we control this? How do we in general control these cycles right here, just these? What does our body produce in response to eating breakfast? We make insulin. We make insulin. So this is a hormone. So we have hormone regulation, right? What do we do when we're fasting? What's our body making? Glucagon. Right? So we have enzymes that are regulated by hormones. So the primary mechanisms that all metabolic flux is regulated by are either our levels of enzyme activity or levels of substrate. So talk to me first about enzyme activity. Let's come down and let's talk about enzyme activity. How do we control enzyme activity? You got to remember this. Bioavailability. What is bioavailability? What is that regulated by? Gene expression. What else? So we'll say cellular location. Okay, what else? Protein turnover. What else? I would say gene expression, protein synthesis, protein synthesis, right? You can kind of combine those two. So protein degradation is protein turnover. That's good. There's another one though. It's not bioavailability. How else can we control enzyme activity? What is that? So covalent modifications will alter what? Catalytic efficiency or catalytic activity. So catalytic activity is regulated by, what did we say? Covalent modifications. What else? Like I could phosphorylate or dephosphorylate something, that's covalent. But there's something else that I could do to a protein to change its catalytic activity. So inhibitors is one way we can decrease, right? But we can also increase. Can we come up with one term that would cover all of them? I would say, so usually it is allosteric, not always, it depends on the inhibitor, right? But I would say binding of regulatory molecules. Right, that could be positive, that could be negative, right? It could be anywhere. So that's a lot of different ways that we can regulate just an enzyme, right? And that's only half the picture. What's the other half? What else can we use to regulate flux? Enzyme activity and what? Substrate availability. Okay, let's talk about substrate availability really quick. How do you get substrates? It's the most common way we get substrates. Intake, we consume them, right? So consumption of substrate. So you could say that's your diet, right? In the example above, how did we get glucose? We either ate it or what? We generated it from a storage, right? So that's the other way that you can get it. So you could say released from storage in the body. And you could say organism, right? Well, but you have to have something else there. Like if you're going to make glucose from pyruvate, you have to have pyruvate there. You're not really making it from scratch. You always have the building blocks available. right? Okay. So now we understand really these levels of enzyme activity and levels of substrate. It's really pretty complex. Okay, so now when you look at all the metabolism that happens in the body, it can be a little bit overwhelming. Yes, it looks like the New York City Metro. Yeah, it looks pretty crazy. So this links all of our substrates to products using a grid, right? And it's color coded depending on what pathway you belong to and all this kind of stuff. But what you kind of notice, if you just generally look at it, now you're not going to have to memorize anything like this because this is really insanely crazy, is that you'll see that many things are linked in many different places, right? So it's important to keep in mind the forest when we stop and look at a tree, because that's what we're going to do. We're going to stop and look at a tree that is glycolysis first. But you have to remember that that tree does not exist by itself. It exists in a forest, and everything in the forest is interconnected, right? So don't lose sight of that, but don't get overwhelmed by it. So easier said than done, right? But that is just too much detail for us. Here's what we're going to study, right? This looks a lot better. So when you look at this, these are our major, this is the most basic metabolic map that we can have that's going to look at 4 major biomolecules. So if we look here at the very top, right? Proteins, nucleic acids, carbohydrates, and lipids. What do we call these? These are 4 what? Major macromolecules. Okay. Now, when we are generating or using these 4 major macromolecules, because what do you notice about those arrows? Most of them are what? One-way or two-way? They're mostly two-way, right? So we notice that we're going to share lots of intermediates, right? You see how they're linked together? So all of those things, right, are similar. So if we look down here, do you see these in red, right? These are 7 small biomolecules that are involved in the different pathways. So these are very, very common things. ATP, right? That's super important. We know that one already. We have to have water, right? And cellular respiration and photosynthesis, all that kind of stuff. We need oxygen, we need water, we need ATP. We're going to either produce or consume CO2, depending on if we're in cellular respiration or photosynthesis. Now, what about NADH and FADH2? What are those two? They're coenzymes that are actually going to be electron carriers. So they're going to be used in redox reactions, which are going to be really, really important, really important. And then we have some primary metabolites. And our primary metabolites are the ones that are in blue, all these blue ones. So it's kind of the middle of the table. We have major macromolecules, we have primary metabolites, and then we have small biomolecules. And we can kind of go in any direction, up or down on these pathways. So we're going to focus in first to start with on carbohydrates. Okay, so if we take this map and we overlay on top of the map the pathways that get us to those different points, what you're going to notice is that we can group these pathways into 2 major classes. So we can have what are called energy conversion pathways. So our energy conversion pathways are the ones that are red here. I guess I shouldn't do that in the blue. I should do that in the red. Energy conversion. Okay. The blue pathways are different in that these regulate synthesis and degradation. of all of our 4 major molecules, right? Proteins, carbs, fats, all that kind of stuff. So you got to think that there are really 2 classes. Either you're synthesizing and degrading things, or you are converting energy from one form to another form. They're 2 different things, okay? So what we're going to focus on to start with are these energy conversion pathways. Take Biochem 2 if you really want to see regulation of synthesis and degradation. It's really awesome. Okay, so when we talk about whether a reaction, an individual reaction within these pathways is going to happen in the cell or not, what are we worried about? What have we worried about since, I don't know what chapter that was when we started talking about delta G? Free energy, right? What does delta G have to be for the reaction to go inside of the cell? Must be negative. It has to be spontaneous. So when we look at our formula for delta G, the problem with this formula is that we're assuming the concentrations of reactants and products are at equilibrium. Is this true? No. You're going to reach equilibrium when you die. And we're really talking about a living organism, so we're not at equilibrium, right? So that's one problem. And the other thing is that we can couple reactions. Reactions don't happen by themselves inside of the cell. If we couple reactions together, then we can look at the overall delta G of multiple reactions put together. But what do you have to be coupled? What's the definition of a coupled reaction? We've talked about this. Well, how do we know that they're performing together? So I'm going to tell you that these 2 reactions are coupled reactions, these 2. Phosphoenol pyruvate going to pyruvate, and then ADP going to ATP. No. Shared intermediate. The product of one is a reactant of the other. That's the definition of a shared intermediate. So what we're doing here is we're harnessing the energy that is released when I convert phosphoenol pyruvate to pyruvate, because that's negative 62. And I'm going to use some of that energy in order to phosphorylate ADP to get ATP. So this is a reaction where I'm harnessing the energy, I'm capturing the energy and transferring it to ATP. So because these 2 are coupled, I have a negative delta G, right? So the other way can happen as well, right? I know that ATP hydrolysis, if I go in the opposite direction, has a delta G value of what? If I take this reaction and I go in the reverse direction, delta G is equal to negative 30.5, right? So if I take that in the reverse direction and couple it to a reaction that has a positive delta G, I can drive that other reaction. So you can use it either way in the cell. Okay, now the other thing we talked about is substrate concentration, right? If you have a very low product concentration, remember we talked about the shift of a reaction, right? Le Chatelier's principle. As you deplete the product, the reaction will shift to the product, right? I know it's Monday. This is tough. So what we can do is if we have a large multi-protein complex, that means that enzyme 1, right, converts A to B. Enzyme 2 converts B to C. Enzyme 3 converts C to D, and so on, right? So as soon as we make B, we're going to use it. We're going to deplete it. So then what's going to happen to A? More of A is going to get converted to B, right? So that's another way that we can drive the reactions to change delta G. So we can look at the math and we can prove that that's true, right? So we know we're looking not at delta G, not biological standard state delta G, right? We're looking at actual delta G. This is actual. right? Actual. We want to actually look at the actual concentrations of our two substrates. So if we look at a reaction where we convert glucose 6-phosphate to fructose 6-phosphate, if I look at the standard free energy, it's a positive 1.7. That would never happen, but it does. So how does it happen? Well, as soon as glucose 6-phosphate is turned into fructose 6-phosphate, look at the concentration difference. Do we have a lot of fructose 6-phosphate floating around? No, because we're going to use this in the next reaction. We deplete this. This is low concentration because it's used in the next step of the pathway. Right? So if you plug that in and you calculate delta G, actual delta G with the actual concentrations, now what do we have? Negative 2.9. So now is this reaction spontaneous? Yes. Is it going to occur? Yes, absolutely. Right? So there are different ways that the cell can use conditions within the cell to drive reactions, right? So now that we kind of know how reactions happen, if we're going to talk about glycolysis, we have to understand sugars. Y'all talked about sugars in organic very much. Not really. Yes? No? Just a little bit? Okay. So we're going to talk about monosaccharides, we're going to talk about disaccharides, and we're going to talk about their structure because it's really, really important. So glycolysis comes from two words. Comes from glycolys, Glykys, which means sweet, right? It's W-E-E-T, sweet, not sweat, sweet. W-E-E-T. Oh my goodness, this pen. Giving me problems today. And lysis, what does lysis mean? Break, to break. So what are we breaking? We're breaking the sweet. We're breaking up sugars. So when we talk about simple sugars, we're going to talk about two kinds of monosaccharides. We're going to talk about aldehydes and we're going to talk about ketones. So if we have an aldehyde inside of a sugar, we call it an aldose. If we have a ketone inside of a sugar, we call it a ketose. What part of the word tells you it's in a sugar? The ose. The ose tells you sugar, and then aldehyde and ketone, right? So if we look at two examples of simple sugars, glucose and fructose, what is the molecular formula for glucose? C6H12O6. What's the molecular formula for fructose? C6H12O6. They have the same molecular formula, but are they the same? No, absolutely not. One of them's an aldehyde, one of them's a ketone. So we can't just use molecular formula. We're going to have to use other designations in order to identify them and to be able to know how they're going to be used in metabolism. So you know that not all sugars taste as sweet as each other, right? So what do we have in our cabinets at home? Sucrose. Sucrose is a disaccharide, right? But when you read food labels, what is the most common sweetening agent? High fructose corn syrup, right? You've heard that before. Why in the world on those ingredients do we have fructose, but when we bake, when we cook, we use sucrose? Everybody know? Fructose tastes about 1.5 times sweeter than sucrose. So manufacturers will mix in fructose with the sucrose in order to use less ingredients and make something taste just as sweet. So that's why you see high fructose corn syrup on lots of the labels, because it's very economical for large businesses to use more fructose than sucrose. Isn't that kind of interesting? Things you never knew you wanted to know, right? Okay. So when we look at simple sugars, they actually exist in two different forms. They exist in a linear form and they exist in a cyclic form. So have y'all talked about that reaction before? Converting it from linear to cyclic? Yes? Some people say yes, some people say no. If you tell me no, I'll go over it in more detail. If not, I'm going to assume you know it. No. Okay. That's okay. So when we look at any given sugar, right, you have to be able to first number the sugars so you know what's going to happen. So you want to number the sugar so that your carbon, your carbonyl carbon that's in a double bond with the oxygen gets the lowest number possible. So if we look at the first one, we look at glucose. Glucose is an aldehyde. If you remember the difference between an aldehyde and a ketone, what I remember is an aldehyde has only one R group. It's all alone. But a ketone, like a lock and key, how many R groups do you need? R1, R2. We need 2 R groups. So ketones, lock and key, aldehydes all alone. Okay. Ooh, trying to erase. Okay. Oh my gosh. Problems today. So when we look at these, you have to pay really close attention to your carbonyl carbon. So in an aldehyde, it's going to be carbon number what? All the time in an aldehyde, it's always going to be carbon number 1. In a ketone, it can vary, but usually it's carbon number 2. So when we do this, when we convert from the linear form to the cyclic form, what's actually going to happen is you find the chiral carbon. Y'all know about chirality? Okay, so what is chirality? 4 different substituents, right, on a carbon. So how do I know if I'm just looking at these that these are chiral or not? If you have something that's a plus sign, it's chiral. They do this, they call these Fisher projections, and that's how they draw them, so that you can really quickly look at it and how many chiral carbons are in glucose. 1, 2, 3, 4. We have 4 chiral carbons in glucose. How many in fructose? Isn't that so nice and easy? Like you don't really have to analyze it. So it's one of those things that's very helpful. So when we look at glucose, if you find the chiral carbon that is farthest from the carbonyl carbon, what number carbon am I? Chiral carbon farthest from carbonyl. What carbon am I? Carbon number 5, right? This is my chiral carbon farthest from the carbonyl. Look at the hydroxyl that's attached. When we do this, this is the oxygen that is going to be in the ring. So do you see this oxygen here? That is this oxygen here. This oxygen is very, very important. When you look at it in the Fisher projection, if it's on the left, we call it L-glucose. If it's on the right, we call it D-glucose. So what is this that I'm looking at here? L-glucose or D-glucose? I'm looking at D-glucose. And D and L, you know, it sounds really simple, but the D and L is super specific because y'all have studied enzyme mechanisms. How important is the 3-dimensional arrangement of your substrates in the enzymatic pocket? Incredibly precise, right? It has to be perfect. So you cannot metabolize L-glucose. If you were fed a diet of pure L-glucose, you would starve to death. But D-glucose is cool, right? That's how precise it is. Same thing with amino acids. You have D and L in amino acids as well. Yes. Yeah, I think so, because it just has to activate the receptor. It doesn't have to undergo an enzymatic change. It just has to bind to the receptor. So it should, yeah. All right. So if we look at this, right, we have D-glucose. In order to make the ring, what's actually going to end up happening is that this straight molecule is going to fold in on itself. So the two ends, what is that? I know, that's crazy. All right, so we have this straight chain, right? Think of the chain as like a shoelace. The two ends of the shoelace are going to come together, but what are the two ends going to be? The two ends are going to be that oxygen on carbon 5 is going to go in and create a bond with carbon 1. Now, that's too many bonds for carbon 1, right? So what's going to happen? It's going to have to lose a bond. It loses at the double bond, right? So it turns into a single bond. That's terrible. It turns into a single bond. I swear, this board, it turns into a single bond. You just have to believe me on this. But now, is the oxygen happy? Nope. But tell me down here about this oxygen on carbon 5. Is it happy? No, it's actually got too many bonds, right? Oxygen only wants to make 2, but now it's bonded to carbon 5 and it's bonded to carbon 1 and to a hydrogen. So what's going to happen to that hydrogen? It's going to come off. Where's the hydrogen going to go? To the oxygen that was on the carbonyl. Do y'all see how that happened? Okay, now the figure that we drew here is what's down here. Okay. This oxygen is this oxygen. It's bound to carbon 1. So this hydroxyl right here is this hydroxyl right here. Yeah? Yeah? Okay, now, when we turn it into a ring, we no longer say D and L because there is no left and right. So what we say is either it's in the cis configuration and it's beta, or it's in the trans configuration and it's alpha. So how do we determine that? Well, we have to look at that hydroxyl. So we look at the hydroxyl and we look at the carbon that is outside of the ring. Standard notation tells us that we are always going to draw that carbon going up. It doesn't actually matter, but we always say it's up. You'll find very few examples where it's not. So you look at carbon 6 and you look at the hydroxyl that is now, instead of calling it the carbonyl carbon, now we call carbon 1 the anomeric carbon. So carbon 1, that was and when you get to the top, this pen doesn't like to work. The carbonyl carbon becomes the anomeric carbon. So when we're in a ring, this is the anomeric carbon. So how do I know it's the anomeric carbon? It's bound to 2 oxygens. All anomeric carbons are bound to 2 oxygens. So you can always find it. Now, is this in the cis or the trans configuration? This one is up. This one is down. So it is trans, and we call that the alpha anomer. Because it's going to depend on the hydroxyl on the anomeric carbon, we call it either the alpha anomer or the beta anomer. So have y'all gone into this detail? on sugars before? No, okay. All right, so let's look at fructose. The same thing is going to happen to fructose, where you have the hydroxyl that's on the chiral carbon farthest from the carbonyl carbon. The oxygen is going to bind to carbon 2 now. When it binds to carbon 2, you lose the double bond, and the hydrogen migrates up and attaches to this oxygen. So now what we get, instead of a 6-membered ring, do you see how we have 5 carbons and number 6 is the oxygen? So this is a 6-membered ring, so we call it a pyranose ring. We're actually going to only get a 5-membered ring. So anytime you have a ketone group, even though they have the same number of carbons, you're going to have 1 less carbon inside the ring, because this second R group is going to end up outside the ring in addition to carbon number 6. Okay, so now, what numbered carbon is the anomeric carbon on fructose? Carbon 2 is the anomeric carbon. And this is going to be really important when we name glycosidic bonds, to understand where is the anomeric carbon. So how do I know, if I don't see the Fisher diagram at the top, how do I know that that's the anomeric carbon? It's bound to 2 oxygens, 1, 2 oxygens. Is this the alpha form or the beta form, and how do you know? It is alpha. It is the alpha anomer. How do you know? So we look at the hydroxyl that's bound to the anomeric carbon, and then we look at the greatest numbered carbon that is outside of the ring. Are we looking at carbon 1 or carbon 6? Carbon 6. So compare carbon 1 and carbon 6. Carbon 6 is up, hydroxide is down. So it is alpha. So you have to be able to look at these sugars and identify this stuff.
Can you explain again how you knew to look at those two specifically?
Because those are the rules. Those are the naming rules. So it all depends on whether that, because okay, if you think about this, if I'm going to take these monosaccharides and I'm going to turn them into disaccharides, where am I going to link them? I'm going to make an O glycosidic bond. What's the O? The oxygen right here. I'm going to link this oxygen to another sugar, right? And I'm going to lose the hydrogen. So if I want to know what that glycosidic bond is going to look like, I need to know what kind of anomer I started with, because that hydroxide is going to determine the conformation of the glycosidic bond. Yeah? So if you're trying to find out if it's an alpha or beta anomer, you look at the hydroxide that is on the anomeric carbon, and you look at the highest numbered carbon that is outside of the ring. If they're both up, they're beta. If one's up, one's down, they are alpha. Okay?
So anomeric just means that they're bound to 2 oxygen. And
Yes.
then alpha and beta represent whether there's a transformation.
Yes. Yes. Okay, y'all ready to go on? And we'll look at some examples. Okay, so here's our Fisher projection, right? Our Fisher diagram. We said it could be either D or L, depending on the location of the hydroxide group on the carbon, we call it the penultimate carbon. But what did we say the penultimate carbon is? It's the chiral carbon, what? Farthest from what? from carbonyl carbon. So immediately you go here, right? Okay, and I look and I say, is it on the left or the right? If it's on the right, it's equal to D. Now, if I want to, can I do this? Is it gonna let me? Okay. If I want to draw, whoa, I don't know what I'm doing. If I want to draw L-glucose, how do I draw L-glucose? Okay, so let's see. If I do this, there's my oxygen, right? I have carbon 2, 3, 4, 5. If I flip, I know in order to be L-glucose, I need to have my hydroxide on the left. Can I leave everything else the same? No. D and L are mirror images. They're actually non-superimposable mirror images. So it's like your left and right hand, right? If I took them, can I superimpose my left and right hand? No, it doesn't work. It doesn't work because they're mirror images of one another. It doesn't work. Same thing with your feet, right? So if I drew L-glucose and I had to swap my hydroxyl on the penultimate carbon, what do I have to do with everything else? Everything that was on the left is now on the right. Everything that was on the right is now on the left. So you can put things that aren't chiral in because it doesn't really matter. If it's not chiral, it doesn't really matter. What matters is the parts of the molecule that are chiral. So anything that was on this side is now going to be on this side. So I'm going to do that. Can I write this? No, I cannot. Why not? This hydrogen cannot be bound to two things, right? So be careful about that. Okay, and then I just take everything that was here and put it here. Okay, so H, OH, H, H. These are called enantiomers. Non-superimposable mirror images. These equal to enantiomers. E-N-A-N-T-I-O. Enantiomers. Sorry, some of these things I have to spell out loud. So D and L of the same sugar are called enantiomers. And they're non-superimposable mirror images. But that's not the only variation that you're going to see in sugars. In sugars, you're going to see examples where only one of those chiral carbons has flip-flopped, basically, right? So look at D-glucose and D-mannose. How do you know they're both D? The penultimate carbon has a hydroxide on the right side. So they are both in the D configuration. But if you look at carbon 2, what do you see? OH and H are flipped, right? So we call this a carbon 2 epimer. So D-glucose is a carbon 2 epimer of D-mannose, or D-mannose is a carbon 2 epimer of D-glucose. You could say it either way, right? So how about D-ribose and D-xylose? These are what? Carbon 3 epimers. Are they both D-ribose and D-xylose? Yes, because OH is on the right side. Okay. All right. So now, really where you're going to see sugars is in the cyclic form in the cell because they're more stable in that configuration. So here we have our D-glucose, right? You have less than 0.05% of it in the chain, the straight chain formation. So we have this oxygen, right, that comes up, forms a bond with the carbon, and that carbonyl carbon turns into your anomeric carbon. You lose the double bond, and the hydrogen from here migrates to this one. And now we have two different things that could happen. We can have that hydroxide facing down, like we showed before, or we can have that hydroxide facing up. So we have to really say what's going on here. So if you look at the name, alpha, what does alpha tell you? You're in the trans configuration, up and down. So that's alpha. D-glucose tells you that in the linear form, hydroxide's on what side? On the right side. And glucopyranose is telling us that it's in the cyclic form, right? It's in the pyranose ring form, and it is a sugar, and it's the sugar glucose. So the opposite is if that hydroxide ends up in the up position. So we have an up and we have an up. So this is the cis configuration, so we call it beta. But from the linear form, the hydroxide on the penultimate carbon was still on the right. So we still call it D. It's glucose. It's in the pyranose form. So we call it beta D glucopyranose. How you feeling about sugars now, right? You got this. You got this. All right. So it's really important to be able to monitor sugars, right? You have to monitor somebody's blood sugar. So one way that you can do that is if the sugar is a reducing sugar. How do you know something is a reducing sugar? If it has a free anomeric carbon. So when I say free, what I mean is that it does not participate in a glycosidic bond. Glyco, glycosidic bond. So how about monosaccharides? Are monosaccharides reducing sugars? Do they participate in a glycosidic bond? No. In order to participate in a glycosidic bond, you have to at least be a disaccharide, right? So all monosaccharides are reducing sugars. So remind me of my my rule, oil rig. Oxidation is loss of electrons. What else? Gain of oxygen, loss of hydrogen. Did y'all learn that? Yes? Okay, so reduction is gain of electrons, gain, G-A-I-N, of hydrogen, loss of oxygen, right? If you don't know that, you need to be able to write that, okay? So what we can do is we can take glucose and we can react it with things like copper, and we can make copper oxide, which converts that copper from a 2+ to a 1+. So tell me what happens to the copper. The copper is what? If you go from a +2 to a +1, what are you? That's your reduction. So how do I know the sugar is being oxidized? Okay, if I didn't tell you that, I'm going to look, and what changed here? My hydrogen and oxygen. So I lost a hydrogen and gained an oxygen, so it must be oxidation. Okay, it won't always be given to you, so you have to be able to do that, right? Okay, so there are lots of different ways that we can do this, and there are some new ways. Your book goes over how you test blood sugar using reducing sugars, right? And so you can kind of look at the new ways. But that's a good place to stop, and we'll talk about disaccharides next time. Yep. The free anomeric carbon does not participate in a glycosidic bond. We did pretty good. We're almost done with that section. That's great. If you have questions on sugars, let me help you now. Have a good rest of your Monday. Anybody on Zoom need anything?
Have a good day.
You too.